Hi, everyone. Good evening. Nice to see you. Right. Super. Thanks for logging in. We're going to give it just another minute or two to allow folks to sign on this evening. Hello, welcome. All right. Hey, Michelle, Tom. Thanks for coming, everyone. We'll get started in just a minute or two. Okay. Okay, I'm really excited to see folks. We had a lot of registrations for this evening's program. I was sort of hoping it would be pouring rain at this point, so everyone would be coming in from their gardens, but perhaps some are still stuck out there in the weeds. Okay. Well, in the interest of time, because we've got some great talks this evening, I'm going to get started here. Would probably have some more folks rolling in as we get going. But thank you so much for joining. I know there's a lot of familiar faces here online this evening, but some new ones as well. So welcome. My name is Liz Kreidinger. I'm the steward and executive director of the Cayuga Lake Watershed Network. We're a regional nonprofit organization dedicated to the protection of Cayuga Lake and its watershed for future generations. And our work involves a lot of things, but what it comes down is education, community engagement, and advocacy. And this program is part of a regular series that we have twice a year, which is our community conference series, sometimes in person, sometimes virtual. But we like to bring together folks from across the watershed to talk about interesting or pressing issues related to lake and watershed health. And then also just stories that celebrate the ecology and help you understand more about the beautiful and complex system that is Cayuga Lake and its watershed. So this evening, we've got a program. Let's see, I have to advance my slide. There we go. I had the speakers test out advancing their slides, and of course I didn't test mine after you guys all joined. Okay. So this evening's program is going to be really lake focused, but I just for a moment want to highlight the breadth of the watershed that we work across. So our challenge and opportunity, of course, is that all the land that drains down into Cayuga Lake covers a whole 870 square miles, 7 counties, 50 municipalities. There's 95 miles of shoreline, and about 140,000 people at this point. We just have a recent update on the census numbers of that growing population. And a lot of values and interest related to the health of the lake and the watershed, which is part of what makes this work so exciting and an opportunity to bring folks together with both shared and varying values around how we see the health of the lake. So that's the context. And I just want to quickly highlight that if you're interested in learning more about our work, we do publish 2 full-length issues of Network News, and we recently completed our winter-spring edition for 2026. So that is available online under our resources tab on our website, freely to everyone, and also is sent as a hard paper copy out to our members to enjoy off-screen this summer for your summer reading. So please take a look at that if you're interested in learning more about both what's going on in the watershed and about our own programs and activities as an organization. Okay, and just I want to highlight, who we are is made possible because of volunteers and members, including businesses and organizations who are with us in this work and getting out in the watershed and participating in programs throughout the year and taking actions to steward our watershed. And then, of course, engaged community members like you, who are showing up and interested in learning more about the ecology of the system and how to protect it. So thank you for being here and being part of this. And without further ado, I can introduce our first speaker. So Emily Zollweg-Horan has been working in fisheries research and management for over 30 years. First, she was an undergraduate at Cornell working in the Adirondacks during the summers and picking lab samples during the winter. Then she worked as a graduate student on Montana for the state agency there. She then spent over 5 years working for the U.S. Fish and Wildlife Service in the Great Lakes before she started her career with DEC in 2005, and now she is an aquatic biologist with the Division of Fisheries and Wildlife for the Bureau of Fisheries at DEC. And I will just share one thing that came in through her bio, which was a quote that I did really love that says she's been lucky enough to work with all sorts of cool fish and really cool and beautiful and accessible places, as well as convenient places in her career. But luckily, Cayuga Lake is both beautiful and convenient with lots of fishy experiences to explore. So we're excited to hear about some of that work this evening, Emily, and we'll turn it over to you. So we'll just take a second so Emily can get her slides shared for this talk.
All right. So, oh no, that didn't work. Hold on. Let's try this again.
That's close.
All right. This is annoying me. It worked before.
So we can see the screen, just the plain old PowerPoint screen. I think if you do the share at this point, it'll work. Yeah.
Okay, now you've got my screen?
Yep, we can see your interest
Or
slide now.
my slide? Okay, perfect. So thank you for the great introduction. I'm here to talk about Cayuga Lake's fisheries data and management strategies. I've been working on Cayuga Lake for the last 15 or so years, so I've got some perspective, but our actual management plan goes back to the first days of the DEC, to the early 70s. So there's some cool stuff here. Well, first, a little bit about the Bureau of Fisheries mission, which is to conserve and enhance New York State's abundant and diverse populations of freshwater fishes while providing the public with quality recreational and angling opportunities. So kind of what that means is that it's our responsibility to decide which fish species are going to be the priority in any given water body, and then try to control their numbers either through stocking or regulations or permits, and help the public and other agencies and our other partners keep that water and that habitat in good shape, and also help landowners with their needs. So it looks like a simple statement, but there's a lot going on there, and it makes our job kind of a little bit different, something a little bit different every day, which is part of what I really love about that. Um, so from my perspective, that looks like sampling the fish, investigating fish kills, habitat protection, working with those landowners, like I mentioned, making sure that there's public access to water bodies, um, providing input on fishing regulation changes and stocking numbers, and also coordinating our volunteer angler project, which I'll talk about some more later, and also outreach and education activities. So this is one of those outreach and education activities, getting to talk to people in the community about what we do, and helping them to understand what we do and why. So one of my favorite parts of this job, actually, and one I don't get to do often enough. First, another little look at Cayuga Lake. Liz mentioned it's a pretty big watershed, and it is probably one of the largest of all the Finger Lakes. It's the second largest actual lake at about 43,000 acres. It's deep, over 400 feet, and there are over 140 tributary streams, which a very few of which are actually shown on this figure here. Most of those tributary streams only run when it rains, um, and they have very steep drops down to the lake. Their actual accessibility for lake fish is sometimes only a couple of feet. The bigger tributaries have several miles, like Seneca River, Yoggers Creek, and Cayuga Inlet all have several miles of accessible habitat. Fall Creek, Cascadoa Creek, 6 Mile Creek are all kind of in the 1 to 2 mile range. Taganet Creek is about a quarter of a mile. Salmon Creek is 2 or 3 miles. Um, so those are some of the bigger tributaries that you'll hear me talking about a little bit more as the talk goes on. And I will also mention that it really very seldom freezes over. This past winter, we had a really nice shelf of ice down by Stewart Park at the south end. But that's kind of rare these days. That was actually ice fishable. It was over enough water that people could actually fish through it. Usually it's only the mud flat that freezes, and that's not as much fun in the winter. Our fisheries community is really, really diverse. I'll start with the cold water fishery. Those are what we focus most on with our fisheries management program. So it's rainbow trout, brown trout, lake trout, and landlocked Atlantic salmon. Those are all fish that we raise in hatcheries and stock. The lake trout and the salmon are probably well, yeah, they're the only ones that are native to the lake. Brown trout are native to Europe, but we've stocked them throughout the US. And rainbow trout are actually native west of the Rockies, not east of the Rockies. So we have transplanted those and added them to the fishery. Fortunately, those four species occupy slightly different habitats and provide different opportunities for the anglers. And so it works out. The warm water fishery, we have black bass on here. It's primarily largemouth bass. There are some smallmouth bass, including a recent state record fish, but it is primarily a largemouth bass fishery. There are also northern pike and chain pickerel, which Their populations are somewhat reduced at this point. The development activity in the city of Ithaca has removed most of their habitat at the south end of the lake. So at this point, they are primarily a North Lake fish, where they can spawn in the weeds at the north end, up by mudblocks. We also have yellow perch, sunfish, crappie, um, a nice variety of things to fish for from the dock, or from a small boat. Their forage base is a mix of native and non-native species. Primarily for the cold water fishes, it's alewives. There used to be Cisco in there. We don't think there's any left at this point. The sculpins remain part of the food diet. Rainbow smelt, their numbers are greatly diminished and they're a minor component of the diet at this point. But those were an introduced species. Another introduced species is the brown goby, which has been there for just over a decade now, and has become incorporated into the diets of lake trout and bass in particular, but other fishes as well. And that changes the nutritional status of those fishes. And it also, as the brown goby populations continue to grow, it also impacts how many eggs of those species hatch and how many of those young fish survive, as the round gobies are fairly aggressive egg and young fish predators. Other fish in the lake, lake sturgeon, we're working on a recovery project for lake sturgeon statewide that's been going on for 25 years or so now. We're transitioning from one recovery plan to the next recovery plan, which sounds kind of okay, you just keep going, that's fine. But it's actually a huge milestone for us because we have actually kind of checked off the boxes where we can say that most of our population statewide are actually recovered now, and we're just working on the last two, which is pretty exciting for us. And then the last fish I'll mention is walleye. They were stocked in the lake briefly as an experiment, some 40 some years ago, and there is a small run in Fall Creek in the spring, but it's a pretty limited fishery. It's not ideal habitat for them. They haven't taken off. This is kind of the nuts and bolts of our management strategy in one slide. So yeah, I'm done now. No, kidding. We stock quite a few salmonids, about 70,000 small lake trout in the fall. The Atlantic salmon are split between lake stocking and tributary stocking, as are the brown trout. And the Finger Lakes wild rainbow trout are stocked just in the tributaries and not in the lake. We also stock lake sturgeon. Our fish survey portion of our program is kind of every 4 years we go in and gill net for lake trout, and we do a forage survey at the same time. We just started doing that. And we're also establishing an electrofishing survey for the bass and sunfish. Lake sturgeon, we do a spawning survey as needed. So I'll explain more about that in a bit. And then annually, we have spring fish passage at the Cayuga Inlet Fishway. Sea lamprey red counts, we do that every year. And we do sea lamprey wounding assessments on both lake trout and rainbow trout every year. Kind of keeping track of where that sea lamprey population is and whether or not we need to conduct a treatment. And then we also want to hear from the anglers. So we do have a couple of different ways of doing that. It used to be a mailed piece of paper survey, but now it's an emailed survey that goes out once every 5 to 10 years or so. Goes statewide to everybody, asking about everything they ever fished for. It's a big survey, but it's really important for us to know what people are doing, what they're willing to spend money on, and what they're interested in. And then we also have this point interview survey where we actually send a technician out into the field to talk to as many anglers as we can. We did that a couple years ago, and I'll present some of the results from that in a few minutes. And then every year we also compile our volunteer angler diary surveys. So those are people who are out fishing on the Finger Lakes, um, and they keep a little log for us. And then at the end of the year, they send those logs in, and we get to see what they caught and how much time they spent fishing. Um, Hopefully, they're checking those fish for fin clips so we know which fish are stocked and which ones are potentially wild or stocked without a clip. Um, and that's a really important program for us. So this is a quick look at some of our stocking numbers. The light gray line at the top is total numbers of trout and salmon stocked. Um, on this graph are the lake trout and the landlocked Atlantic salmon. Um, the salmon are stocked in two parts. We noticed a couple years ago after some surplus stockings and the failure at Adirondack Hatchery, which explains some of the variation in the numbers of fish stocked. But we noticed that when we stocked smaller fish in the tributaries, a couple years later, we had better recruitment to anglers' creels. People were catching more bigger fish in subsequent years. Um, so we're doing an experiment right now where we're putting half the fish in the tributaries as small fish so they have time to feed and imprint and hopefully return to those tributaries as adults to spawn. And the other half we're stocking in the lake the way we always have. It's still pretty early to tell whether that's working or not, but we're hopeful. Um, the other thing I will note here is that we stock a lot of lake trout, um, and you'll see how successful that is as the presentation goes on. But that's actually something we're looking at, trimming those numbers so that we're not stocking as many lake trout because they are surviving better than they used to, and we don't need as many anymore. And then this is the Finger Lake strain rainbow trout and the brown trout. The brown trout, again, same as idea as the Atlantic salmon. We stock some directly to the lake and some in the tributaries, kind of hoping that they'll return to those tributaries and provide a fall fishery, tributary fishery for folks. Um, the rainbow trout, those are created at the Cayuga Inlet Fishway every spring. So they're fish that come run out of Cayuga Lake and up one of the tributaries. And then those fish are actually stocked back out into tributaries throughout the Finger Lakes. So um, that's kind of a nice thing that we get to do. And then the lake sturgeon spawning or stocking rather. We've stocked about 37,000 lake sturgeon in Cayuga Lake so far. Um, it sounds like a lot. In real numbers, adult fish we think we have right now is probably only about 500. Um, because they live so long and because they don't mature until they're 15 to 20, 25 years old, even though there's a small amount of mortality every year, it's a lot of years and it adds up. Um, and so that's what we think we have out there right now is about 500 fish. And that's either looking at the survival numbers over time or looking at our recapture estimate. So um, part of what we need to do here is continue stocking so that we get a better genetic diversity going on out there. So that if they do spawn, they're not spawning with their brothers and sisters. They're um, keeping their genetic diversity going. Um, so that's one of the parts of the new recovery plan is to continue stocking populations to get their genetic diversity up as we go forward so that they can persist on their own for hopefully about 50 generations or more. But that's really all we can plan for practically. Um, and we also monitor their spawning activities. So one year I was walking in Fall Creek and somebody called and said, "Hey, there's sturgeon spawning in Fall Creek." And I happened to be out there that day walking the creek. And sure enough, there was sturgeon swimming around my feet and bumping into my legs as I was walking through. Um, I love getting calls like that. Please make more of those. Unfortunately, the conditions that led to the sturgeon spawning in the stream that year are kind of unusual. We need a big spring storm in order to bring the fish in and provide enough velocity of water for them to actually accomplish spawning. And most springs, we don't get a big storm, and so we don't have enough water in the stream for them to do that. Um, but something to look out for. And please give me a call. This is one of the rainbow trout that we see every spring at the Fishway on Cayuga Inlet. Um, we weigh and measure them, we check them for sea lamprey wounds, and then we do that egg take for the Finger Lake strain rainbow trout. And then we also pass them upstreams, pass quite a few upstream so that they can, well, they all get passed upstream, but some of them get passed upstream without being stripped for the egg take, so they can spawn on their own in the stream um, and provide wild fish for the fishery as well. And they are, Yucca Inlet's actually in really great shape. They do a good job at that. The sea lamprey wounds I mentioned, if you haven't seen a sea lamprey before, this is what they look like. They've got a really weird little head with a round mouth. And they attach onto the side of the fish and then use their tongue to dig a hole in the side of the fish so that they can feed. They are fish, but they're parasitic fish. Um, and I think they're really cool, but they're also kind of a pain because they hurt the salmonids that it's my job to manage and protect. Um, Part of their life cycle is after the little itty bitty little grain of sand sized sea lamprey eggs hatch, their larvae actually filter feed burrowed in the sediments for several years, sometimes 10 years or more. Average is about 3 or 4, but it can be longer. Once they grow to about the size of a pencil, an unsharpened pencil, then they metamorphosize and become parasitic and move downstream to the lakes, um, where they feed for a year or a year and a half before returning to spawn and die. By the time they're moving back upstream um, to spawn again, which is where we catch them at the fishway, um, they've already done the damage that they're going to do. And we're just removing them before they can spawn and keep the population going at a high level. We also have a larvicide, which we can use um, on the larval phase when they're in the filtering, feeding in the sediments. It's very specific just to that phase of sea lamprey and just to that species, so that we can control their populations if we need to. Fortunately, we don't have to do that very often. So this is kind of what our spring data at the Fishway looks like over time. It goes back over 50 years. This year was our 51st year, actually. Sea lamprey numbers have been fairly low for the last several years. This year we had, I think, less than 50 sea lamprey total. And so another small year. And our rainbow trout numbers have been pretty stable, about 1,200 for the last decade or so. So things are at the moment looking pretty good, and hopefully they continue that way. This is what a couple of those wounds look like, and the close-up view of the mouth of the sea lamprey if you've never seen one before. The wound on the side of the fish, towards the top, it looks kind of fuzzy. That wound, they didn't get all the way through to the body cavity. So that wound might get a fungal infection. In fact, it looks like it's got a mild one there, but it should heal up pretty quick, and there will be a small scar there, but the fish will be fine. The wound under the pectoral fin has also, it's closed over, but where it's closed over in the dark spot in the middle there, it's not very thick yet. So that needs to finish healing together and get thicker there before that fish is fully protected. But again, that wound is not infected, and this fish will probably survive both of those just fine. So, and a lot of fish do, but not all, which is why we are concerned about that. Um, I joke sometimes that this is our sea lamprey control program all on one side. Amicetes are those larval filter feeding sea lamprey. And when we think that they're in the larval habitat upstream of the fishway, we go and electrofish for them in the fall and count how many we catch per hour. So that's what those yellow bars are. The red dots are when it was possible for there to be escapement over our Cayuga Inlet, the dam at the fishway. So when there's a big storm and the lake level is high in the spring because they're holding water to prevent flooding further downstream, um, that barrier at the dam becomes less of a barrier, and fish and sea lamprey can both just swim right up over it. And that happened this spring. It also happened last spring. Um, but fortunately, we didn't see reds upstream. They didn't spawn upstream. And when we went back looking for amicetes, we didn't see any. So that was some good news. Sometimes we get lucky, and that's how it works. There are also 4 sea lamprey treatments on here. That's all we've had to treat Cayuga Inlet. And that's the only tributary of Cayuga Lake that has both significant spawning habitat and significant larval habitat. Fall Creek has some really great spawning habitat, but it doesn't have that larval habitats to support those fish. So the sea lamprey that spawn in Fall Creek are wasting their time, frankly. Their little larvae are not going to survive. They're not going to find a place to filter feed. So we count how many are there, so we know what the bigger population number is, but we don't worry so much about going back in there and doing a lampricide treatment because the larval habitat just isn't there to support it, and to make that worthwhile. Only Cayuga Inlet does. So there's been actually 5 times when there were enough larvae up in Cayuga Inlet to be worthwhile for a treatment. But the 2007 and 2011 populations, we didn't treat those. We let that go. And that was a choice that we learned from. So let's see. This is our wounding rates on our fall egg take for lake trout. The Finger Lake strain lake trout that are stocked into all over our Finger Lakes. Those fish are from Cayuga Lake. They used to be from Seneca Lake, but now they're from Cayuga Lake. And the graph on the right there just shows our wounding rates when we've measured it over time. There was a significant gap there where we didn't measure it. Those are pretty good at this point. A lot of the wounds that we see are really well healed up wounds like this one, which probably did go all the way through. And this fish has recovered from it and lived to spawn at Teganic Park. So that was kind of cool. Uh-oh. There we go. Okay. Every 4 years, we do a gillnet survey where we set nets at standardized sites throughout the lake. It's a standardized gear. We set it the same way every time, so that this data is comparable from 1 year to the next. And if you look at the years that we surveyed, it's not actually every 4 years. There was several years there where we did it every year, and then I think we skipped one also. But generally speaking, this is our numbers caught over time. The way these nets are set is very targeted towards lake trout. They don't, because the Atlantic salmon, brown trout, and rainbow trout inhabit slightly different areas than lake trout do, we very rarely actually catch them. And as you can see here, we haven't caught Cisco since 1979. Um, Rainbow smelt have been fairly scarce the last 14 years or so. Um, and yeah, it's been a little tough on some of the prey fish, which is why we started the new forage fish survey so that we can get a better handle on how our prey fish are actually doing. And that survey uses different gear that is specifically targeted towards those prey so that we can um, track their populations over time. We've only done it once so far, so it's a very short data set at this point. So I'm not going to show you that because it's really boring. It's all alewife with a couple of gobies and well, we also caught some surface feeding fish at the same time. So that was kind of fun, actually. Um, our electrofishing survey for bass, we've only done this a couple of times. So this is the most recent survey from last year. The results are pretty similar to the previous surveys, though. But hopefully, as we get more of these under our belt, we'll have a better handle on how those largemouth bass and the sunfish are doing out there. Right now, what we see here is that there are a lot of bigger, older fish, and they're growing really well. They're fat, they're happy. But there are very few small fish. And this survey should pick up those fish. We should be seeing clouds of young of the year and age one fish as we're moving through the littoral zone of the lake. But unfortunately, every time both times I've done this survey, we haven't seen that, and that worries me. That worries me a lot because either I'm just happened to be out there in years when there wasn't a whole lot of those fish produced, or there's something very seriously wrong with either the habitat or predation on those small fish, such that very few are surviving. Um, which means that this population that we see here of large older fish, as they die off, they won't be replaced, and that is definitely of concern. Um, our Creel technician managed to talk to over 1,300 anglers, which I'm pretty impressed by that. She even managed to talk to a couple people who were um, at Cayuga Lake for a tournament and get their um, information on how much time they had spent fishing and what they had caught and what they were interested in. And that was nice to see that. There's a couple spots that show up as big shore fishing spots, kind of as one would expect. Taganic State Park and Mudlock are both popular places for people to shore fish, as well as Cayuga Lake State Park. Um, Dean's Cove won the prize for being the most active site. Um, again, not too surprising. Um, and kind of interestingly to me, as I expected, um, lake trout are by far the most popular. You know, anglers spent the most time chasing lake trout, um, and largemouth bass. But they were also out there saying, you know what, I don't want to just catch a lake trout. I want to catch, maybe a brown trout or a rainbow trout or a salmon. And that's what the cold water combination category is. There were a lot of people in that category as well who were fishing for lake trout, but also maybe doing something a little bit different every now and then, hoping to catch something else as well. And then there's a lot of people who are just out there happy to be fishing, and anything is a good thing. So um, I tend to be one of those anglers, so I can empathize with them. Um, in terms of numbers of fish caught, it pretty much follows the effort, other than the yellow perch number, which is just out of this world, but not too surprising because there are um, a lot of yellow perch out there as it happens. Yes. Um, A lot of lake trout caught, a lot of largemouth bass caught. Um, Summer tended to be the months when people caught more diverse numbers of fish, but the yellow perch fishing carried through the winter. The winter that the creel survey was done was 23, 24, so it didn't freeze that winter, but so the boats were out at the north end all winter long, and they were very successful at catching yellow perch. Um, the brown trout, Atlantic salmon, and rainbow trout numbers here aren't super impressive as raw numbers, but as catch per hour, fish caught per hour by people targeting those species, they're actually pretty comparable to similar numbers calculated for Owasco Lake and Skinny Atlas Lake. So um, people are actually doing fairly well on rainbow trout out there, brown trout and salmon as it happens, at least compared to the other Finger Lakes nearby. Our angler diary participants, again, catch a lot of lake trout. There's a refrain there, right? We catch a lot of lake trout, and smaller numbers of the other species. Our management goal is actually to have a more diverse fishery, which is why I mentioned possibly why we're trying to trim the number of lake trout stocked, because their numbers are so overwhelming that it is really easy to fish for them. And for people who are targeting other things, maybe they're having a harder time because um, either competition with the lake trout or just just having a hard time catching something other than a lake trout. So we're trying to trim the numbers of lake trout with the better sea lamprey control the last decade or so. The lake trout survival is doing really well, and our stocking numbers were based on a poorer survival. So we have to adjust how many fish we stock based on the new survival numbers, which means stocking fewer fish to have the same population that we have all along. The other thing that I'll note here, these are angler caught catch per hour on largemouth bass and smallmouth bass. And if you look at the last decade or so, it actually kind of looks like things are declining. And I don't know if that's real or not because our participation is so low in that program. Um, the Angler Diary program has been around since 1984 or before, I think, in some lakes. And that's a long time. And our anglers are getting older and retiring out of the program. And we're not doing as good a job at recruiting new anglers to the program as we should. And so what we have here is sample size bias, where there's so few anglers participating in the program that just a few anglers having a good year or a poor year actually bias our results in one direction or the other. Even our lake trout data, our cold water graph there might be biased by um, some people who are just really, really good at catching lake trout. Um, so this is my plug for if you're somebody who's interested in fishing and willing to keep a little logbook for us and send it in at the end of the year. You'll get a new logbook, a fresh one for the new year, along with the report on what was caught the year before. And it's a really nice way to contribute um, to the program and let us know how things are going out there. So in case we do need to make a change, like stocking fish or fewer fish or change a regulation, this is a big piece of our decision making process. And then this is my last slide. These are the pictures I like to see in my inbox. Happy anglers. Our lake trout population, as I mentioned, is kind of doing maybe a little too well. But those other salmon species are also doing well compared to the other Finger Lakes. Our largemouth bass anglers seem especially happy with Cayuga Lake. Um, And I'm really hopeful that it's not short-lived, that there's something I'm missing in terms of where those young of the year fish are, um, so that we can keep that going for them. Lake sturgeon recovery is also going really well so far, but there's more to do there. Um, and with that, I'm going to say thank you for your time this evening. I really appreciate y'all showing up. And I hope you have lots of good questions for me, because that kind of makes my day go round. Um, Feel free to email me or give me a call if you think of something after tonight's over. Um, or if you happen to see lake sturgeon spawning in the future, make note of my email address and send me an email. I'd really appreciate it. Um, and if you've got my phone number from before, I actually have a new phone number. So make sure you write this one down. And that's it.
Great. Thank you so much, Emily. That was really interesting. We certainly have some time now to open it up to questions. So if you would like to do that, you can unmute yourself. You can raise your little yellow Zoom hand, or your real hand if you turn your video on, and I can call on you. All right, we have Charlie.
I
Go
think it
for it.
Hillary. I think it had Hillary too. I think she was first. Is that right?
I don't see
Maybe
Hillary's
not.
little hand,
All right.
so go
Maybe
go
that
for
was
it.
just a clapping she was doing. Um, yeah, wonderful presentation. Thank you. Couple questions. The sea lamprey. I've always assumed that's invasive. Is that true or not, or is it a something that's always been in Cayuga Lake?
They probably have not always been here. Um,
Mhm.
Certainly at this point, well, at least as of 10 years ago, their numbers were such that they were impacting the survival of the salmonids that we're trying to manage for. And
Mhm.
that impact makes it hard to um, made it harder for us to manage those populations. So that's
Mhm.
why we do control. We're not an eradication program for those. It really is just a control program.
Okay.
So
All right. And why do they release from a lake trout at, you were showing wounds that the sea
mhm.
lamprey release? Is that part of their life cycle that they get to a point where they have to let go, or does the fish knock them off, or some of
The
both?
fish are pretty good at knocking them off.
Hmm.
Yeah. You'll actually see fish kind of leap out of the air and shake,
Hmm.
and the lamprey go flying. As soon as we pull a fish out of the water, usually a lamprey will drop off if they're attached to it, because they like to be in the water too. That's how they
Okay.
breathe. Um, Sometimes the fish is dead and they drop off that way. um, I will note that sea lamprey are very specific to cold-blooded things. So especially when I'm talking to kids, if they're going to go out swimming in the lake, as long as their lips aren't blue, they're probably
Hmm.
okay. They're safe from sea lamprey. When they get really cold, though, those sea lamprey might mistake them for a fish and try to attach. So
Hmm.
it's easy to pop them off with you get your thumb under them and just pop them right off, but um, it might be a little uncomfortable for a couple minutes there. So
Yeah, that'd be a little scary. One more topic, perch fishing. So, I sometimes I see all the boats at the north end of the lake, just
mhm.
dozens and dozens at times, and each boat sometimes they bring in, I don't know what the limit is now, 25 or 50 or something. It's a pretty big
I think it's
number.
still 50, but yeah.
50, yeah. And I just wonder if that fishing pressure has any concern on the perch population, or are they just doing so well that there's plenty of perch available?
Um, it is a concern. I will also say that it's a concern that as an agency we haven't tracked very well. We don't have an index for their population. We don't know. We have that snapshot of the creel survey, so we have kind of an idea of
Hmm.
what the effort is and what the impact is, but we don't know what the baseline population is, and
Hmm.
we don't have any way. not tracking it in the angler diaries, and we don't have a survey that targets a population estimate for yellow perch. So it's
Yeah.
something we're not doing a very good job of.
I just anecdotal observation, it just seems like over the last 30 or 40 years that I've lived in this area, it just seems like there's a lot more boats on the north end of Cayuga Lake than there were 10, 15, 20 years ago, particularly in the early spring and such. But
Yeah.
so,
Oh, I
okay,
think you're right.
all right. Well, thank you. Maybe
Sure. Thank
maybe
you.
somebody else will have a question.
Super. Who's next? All right. Well, y'all are thinking about that. I have a few, so I'll share one. The first was I was just curious in that graph that showed the rainbow trout populations that you're monitoring down in the inlet, right? There was like a huge spike in 2015.
Mhm.
I'm just curious what what happened there. Like that uh, was pretty large. Is there an explanation?
Um, no, it was just a really good year.
Okay.
Um, there had been a couple years before that where it was not a good year, where we actually had to go upstream and electrofish. But that was 2015. They had recovered and they were doing better.
Okay. Um, and then my second question is, what do you think can be done, or what would the approach be, or perhaps what is the agency already thinking about to try to better understand the decline in the young largemouth bass population?
Um, so one strategy would be to use a different sampling method, um, and go out at a different time of year and see if we can um, get them that way. So that'll be the first thing we do is we'll try different gears and go out at different times of year. Um, and hopefully we can find something that we can do that's repeatable. Um, so that's going to be the challenge for next year, is to figure that one out. Um, or maybe even this fall, actually. So that we can start building that data set so that we can start noticing when there's trends either down or up. So
Okay, thank you. I see Fisher St. Clair has a question, so go ahead and unmute yourself.
Yeah, can you hear me?
Yep.
yep.
Okay, yeah, first, thank you for giving that talk. It was super informative. I was just wondering with round goby, if there's any like methods to reduce their populations, I guess.
No, not really. The best we can do at this point is not spread them to other locations. Um,
Right.
yes, you can harvest all the ones you catch by accident, but there are so many of them out there at this point, um, that's kind of wishful thinking.
Yeah.
They, like sea lamprey, they collect kind of all in one place. They funnel into one place to spawn. So they're vulnerable in that way. So we can target them because they congregate. Um, Round gobies don't do that. They spawn several times a year and throughout the shoreline of the lake. So there's not a whole lot we can do just to specifically target just the round gobies.
Is that your like leading theory for why there might be less of the smaller bass, like the younger
That's
bass?
part of it. Yeah, that's part of it. Their habitat definitely overlaps, and gobies are known egg predators. So if they're getting into a bass nest and eating all of the eggs, then of course there's no little bass that from that nest that year. Um, and that may very well be what part of what's happening.
Yes, well, thank you.
Yep.
Anyone else? Okay. Well, great. That was super. Thank you for answering those questions, Emily. It sounds like, yeah, if anyone wants to be participating in the Angler Diary program, that Emily is ready to hear from you. And so I hope that talk was also inspiring as you're thinking about getting out on the lake this summer, and getting out and relaxing. So as we're out there enjoying the lake and the fisheries, we also want you to be awares of harmful algal blooms. So I think we'll move on to Alyssa's talk as well. Thank you again, Emily. So Alyssa, if you could come back to us here. What I'll do is I'll just start your introduction. You can work on pulling up your slides while I do that. Great.
Sounds good.
Okay. So Alyssa Johnson is the Outreach and Programs Coordinator and Cayuga Lake Harmful Algal Bloom Program Coordinator for Community Science Institute. She's a lifelong resident of the Finger Lakes with a background in wildlife management, environmental education, and natural resources conservation, which she all brought to CSI in 2024. So prior to that, she has worked with a variety of government agencies and nonprofits, including Fish and Wildlife Service, New York State DEC, and the Audubon Society. And we're lucky to have her because she's managing this really important program for Cayuga Lake, which we're a proud partner in. And I get to work with Alyssa quite a lot in thinking about how we continue to communicate the findings and information that comes out of this program to you all throughout the summer and the entire year, in our advocacy as well. So with that, I will turn it over to Alyssa for the next talk. Thanks, Alyssa. Looks good.
Great. Thank you. And the presentation looks good from your side, Liz? Okay,
Yeah,
great.
we can see your first slide.
Thank you all so much for logging on tonight, and thank you to Liz and the network for inviting Community Science Institute again to present at the Spring Community Conference. This was one of the first events that I attended when I started in 2024. I was not presenting, our executive director did, and it's one of my favorite events of the year because I get to mingle and connect with lots of locals, Cayuga Lake lovers, and share what we've been learning. So my presentation this evening is What Lurks in the Lake, and I'll be talking about the 2025 updates and findings about concerning harmful algal blooms on Cayuga Lake, as well as a few things about this present season. And before I get into the presentation, I wanted to just share this photo. I love it. I don't love the content of it, but this is just a phenomenal picture that was taken last year. Oh, the date is wrong. October 4th has not even happened yet this year. October 4th, 2025, by a local hobby drone operator who has become a friend of mine. And this was taken in Union Springs, in just phenomenal photo. There's a series of photos where he turned the drone to the north and to the south, capturing images, and the HAB just extended as far as the drone could see. So I wish that it wasn't a huge harmful algal bloom, but the photo is really impactful and quite interesting to look at. So I work for Community Science Institute. We are a 501c3 nonprofit and state certified water analysis lab. Our new mission, I guess it's not quite new now, but it's a little over a year old now, to inspire and empower communities to safeguard water resources by cultivating scientific literacy through volunteer water quality monitoring, certified laboratory analyses, and education. We are a nonprofit, but we have a business side to our organization where we offer water testing to community members, to residents, to local business owners, universities, anyone that is mandated or regulated to conduct water quality testing. We are the only water quality lab in Tompkins County, and we're a nonprofit, which is kind of a unique situation. Other water labs that you may learn about or bring your own samples to are often corporate for-profit businesses, and we really pride ourselves on being able to provide a lot of education and time consult with clients coming in with questions. There's kind of three facets of Community Science Institute: our fee-for-service water testing lab. And I want to mention that we launched at the end of 2025 a water testing assistance program that offers a select list of common in-house analyses that we do at 50% to those who identify themselves as needing a little bit of assistance. You can contact me if you want more information about this, or contact us at CSI in general. But we believe that everyone should have the ability to know what their drinking water, the quality of their drinking water is, and we don't want cost to be a barrier. That program is supported by the Rotary Club, Ithaca Rotary Club, and maybe one day we'll be able to offer some testing 100% free of charge, but right now it's 50%. So the fee-for-service water testing lab is one facet, and then we have our volunteer water monitoring programs, synoptic stream and lake monitoring, biological or biomonitoring, and of course the harmful algal bloom monitoring. And then the third facet is our education and outreach efforts that we do through a variety of different avenues. Our Journey of Water Youth Education Series is starting this weekend. On Sunday, we'll be taking a water quality monitoring cruise where we collect water samples with kids, and they're using the equipment, they're learning why we're doing what we're doing. The program basically takes a journey throughout the Cayuga Lake watershed, visiting wetlands, wastewater and drinking water treatment facilities, taking water quality cruises, doing some biomonitoring. It's really, really fun, and I quite enjoy it. We attend many professional conferences throughout the year. We offer other free public events, like in the, the spring, in February, we offered an event called a Data Jam, which was an opportunity for people to come together and explore our database with support from staff and board members. Our water quality database has over 150,000 data points on it that concern water quality, and again, to empower and encourage people to learn about drinking water and groundwater. The database is free and open access to anyone who wishes to view it. And I'll talk a little bit more about that in a few slides. Similar to the network, we also publish two printed, there's a digital version and then get them printed as well. Our Water Bulletin newsletter, which is full of articles written by staff about the year's projects and things we've learned, or trends, or an interesting partnership we may have had. I'm always responsible for writing the article about harmful algal blooms. So there is an article in there that's a bit longer than my presentation will be tonight, covers a bit more if you're interested. And then our annual report, of course, is going over our stats for the year and our finances, our gains and losses. And I make both of these. I format them and design them with help of my coworkers, but these are two proud publications for me to get out there every year. Okay, so to start talking about harmful algal blooms, we on Cayuga Lake are part of a larger statewide monitoring program. In 2012, the New York State DEC began documenting HABs statewide and started some outreach efforts. We received a question someone submitted during registration, and they wanted to know, are HABs a recent phenomenon or something that's occurred in the past 150 years or so? Harmful algal blooms are a fairly new phenomenon. I would say within the last 20, 30 years, these events are happening larger events more frequently, covering more area spatially and temporally. But the organism that causes a harmful algal bloom on Cayuga Lake is cyanobacteria, which is a natural organism. It is one of the oldest organisms on planet Earth, about 3.5 billion years old. And they are found in freshwater all over the world and are not innately harmful. So cyanobacteria has existed in Cayuga Lake for as long as Cayuga Lake has existed, but the blooming phenomenon is fairly new within the last several decades. So the New York State DEC started paying attention in 2012. In 2017, dozens of reports were made from concerned community members around Cayuga Lake. There were several samples collected and confirmed to be harmful algal blooms, and I believe there were 5 confirmed HAB reports that year. In response to that, the Cayuga Lake Watershed Network and Community Science Institute began talking about what was going to be done about this. There are many volunteer monitoring groups across New York State. The different watershed associations are usually the ones who house the program. But because Community Science Institute has a state certified water lab and we are able to process samples in-house, it just made sense for us to work on this collaboratively and cooperatively in getting information out to the public in a timely manner, working together to create different marketing or educational resources. This event tonight is a great example of outreach efforts that we work on together and recruiting volunteers. And we both act as professional points of contact if someone wants more information on a technical level. We are both available to talk with folks all the time, unfortunately, about harmful algal blooms. I wish I had a different topic to talk about, but this is an important one, so I'm glad to be able to help. This chart shows the number of HAB reports over time. So while Community Science Institute has housed a volunteer program since 2018, volunteers are not required or expected to be monitoring every inch of the lake 24/7. So these numbers are representative. What actually happened with the actual number of harmful algal blooms that occurred on the lake, probably impossible to know, but this is a pretty representative year, and I think we can all agree that the problem looks like it's getting worse. Last year was record-breaking in many ways, and the year before that, 2024, was record-breaking, and then we broke those records the year following. So we've already broken one record this year with our earliest harmful algal bloom, I believe, in New York State history. Good for Cayuga Lake on April 23rd. I'll talk a bit more about that shortly. So this is a really collaborative effort. Yes, Community Science Institute is the main, we're in charge of facilitating the logistics, getting volunteers trained, onboarded, assigned, monitoring areas, handling logistics to do with samples and analysis and getting that information out. But we could not do it without the support of our 3 funding government agencies, the Seneca County Health Department, Cayuga County Health Department, and Tompkins County Whole Health Department. All provide funding to help support this program. To my knowledge, no monitoring group concerning harmful algal blooms in New York State receive state funding. Now, some grant opportunity, a grant opportunity has just come out in the last month or so, which could be really game-changing in some ways. But we only receive funding from Seneca, Tompkins, and Cayuga County Health Departments. We work programmatically with the New York State DEC to make sure that we are following their protocol and gathering the data that they need. We work with New York State Parks to make sure that on Cayuga Lake, that where there are swimming areas, that the HABs are being reported accurately and timely, and that we're getting water samples analyzed quickly so that beaches can hopefully reopen safely. And then, of course, Cayuga Lake Watershed Network and Discover Cayuga Lake are 2 local nonprofits that have a big vested interest in Cayuga Lake and help us get a lot of education and outreach out to residents, to visitors, tourists, knowing about HABs. It's funny to me because I feel like everyone knows about harmful algal blooms. And then I'll make a post on Facebook and use the acronym HABs, and almost every single time somebody will come and say, "What's a HAB?" And so that keeps me in check that not everybody is aware and that the outreach is very important and needs to continue. So I really greatly appreciate the support from Cayuga Lake Watershed Network and Discover Cayuga Lake. And then, of course, probably the most important piece of this whole puzzle are the volunteers. We could not do this without them. I'm not sure if this is true, but I heard through, professional network that there are 2 people, or there have been in recent years, 2 people that monitor Honeoye Lake, which is 5 miles long. I cannot relate. We have close to 100 volunteers, and that is not even close to covering all the shoreline, but we do a really good job. There's about 50% coverage, and then lots of concerned community members that are constantly emailing, reporting, letting us know what they're seeing out there. So in 2018, when our monitoring program began, the difference that is between our program and New York State DEC's monitoring protocol is that we collect samples. I wasn't around in the early days of the program, so I don't know. I know samples were collected either initiated by DEC or by DEC, the agency itself. But currently, there are, to my knowledge, are no other monitoring groups collecting samples as regularly as Community Science Institute is on Cayuga Lake. Now, from 2018 to 2024, we asked volunteers to collect a sample with every HAB that they reported. We wanted to amass a huge data set quickly to learn about potential hotspots on the lake, toxicity trends, the genus of cyanobacteria or cyanobacterias that are making up the bloom. And we learned really valuable and important information. And then 2024 happened, and two things. It was a record-breaking year with 127 reports, or 129, and a sample to go with almost every single one of those reports. The logistics of getting samples from around Cayuga Lake to our lab in Ithaca can be challenging at times. We have some people that live an hour away. They report a HAB, they collect a sample at 4:45 on a Friday. They may not want to drive it an hour to and then an hour home to deliver that sample. So we have some really great volunteers that are in a role just to support transport of samples, but it was becoming apparent that it was causing a lot of fatigue, and we wanted to retain volunteers. So in looking at the data, we were seeing that sort of the things we were learning year after year were being reconfirmed. Over the last several previous years, nothing groundbreaking had really come from the data, and repetition is great and very valuable as well. So we spoke with our funders, the county health departments, before 2025 season began, and said, how can we make this more manageable and still give you the information that you need? And so we mutually decided on a more strategic approach to collecting samples, and each county health department identified priority sites in their counties that have some kind of public health implication, whether it be a public drinking water source, it's a regulated swimming area, it's an unregulated swimming area like East Shore Park or Salt Point, where all the locals like to go, but there's no monitoring being done officially by a government agency to make sure the water is safe. So 14 sites were identified in 2025 to make the sample collection and processing more logistically feasible. I was getting ahead of myself, but I will thank the volunteers again. I see some of you on here, and I will thank them again because there's no way that I could do this work alone. So here are the sites from 2025. In Cayuga County, we had, starting from north to south, Cayuga Marine and Outfitters, Harris Park, Frontenac Park in Union Springs, and Long Point State Park. Switching sides of the lake, in Seneca County from north to south, we had the Town of Seneca Falls Water Treatment Plant, Thirsty Owl Wine Company, Sheldrick Point, Cayuga Shoreline, and the Spotted Sandpiper. And then at the south end, Tompkins County, starting on the east side and wrapping around to the west, Salt Point Natural Area, Lansing Harbor, Bolton Point, East Shore Park, and Ithaca Yacht Club. So I've been asked several times, why are there only 4 monitoring or 4 sample collection sites in Cayuga County and not 5 like the other 2? Just because we suggested, why don't you pick 4 or 5 sites per county, and we'll start there. And so they picked 4 in 2025. We have 5 sites identified for 2026 though in Cayuga County. So full 15 for this year. So what did last year look like? We had a little bit of a slow start. In 2024, we received our first HAB report, which was the earliest on record, earliest on Cayuga Lake's record, on June 3rd, 2024. So last year, we did not receive our first report for a full month later. And that whole month of June last year, I was wringing my hands and sweating and like, hello, is this reporting form? Like, we use an online reporting form. Is it working? Is anyone out there? Are volunteers looking for HABs? Like, what is going on? And we just had a late start, I believe, because we had a lot of rain in the spring that kept the lake temperature a bit cooler. And then we had our first report on 7/5, and then a major Western Shoreline bloom event from about Canoga to Ithaca Yacht Club on the 7th, 8th, or 8th, 9th, 10th of July or so. You can see that in that week we received 54 reports. That's a lot. That is a lot. I think in 2024, my busiest day was 12 reports in a day. So this was kicking off the season with a bang, that's for sure. A little late, but really got it going. Quieted down a little bit through July and August, and then the drought really set in. It was very dry, it was very hot, and September and October were not great. And we ended with our latest report on record, which was November 7th, with a persistent HAB that was hanging out in the Teganic Falls State Park Marina for about 2 months. Very small. Probably most people walked by it, didn't even notice it, but we had a very watchful and observant volunteer who kept eyes on it, and I said, keep reporting it as long as you see it, because I kind of like to know how long this thing lasts, and it was about 2 months. So an interesting way of looking at this information is bloom days versus bloom reports. So we could receive many, many reports in a single day, but how many days of the summer did we have blooms reported? And in July, we had 12 days where we received at least one harmful algal bloom report, but there were 71 total reported for the month. 8 bloom days in August, 25 bloom days in September, which is a lot because there's only 30 days in September, and same for October. You can see in September we had 115 reports for the month. So it's interesting to kind of look at how many days. Now, this doesn't mean the whole lake was affected by, if we had one bloom on a given day, that doesn't mean the whole lake is out for recreating and enjoying, but it just means that we had at least one HAB reported on the lake somewhere during that month. So due to our more strategic approach, we were able to collect 34 samples. You can see the breakdown in the table there of how many were taken from each one of the priority sites. Once we receive a sample, the first step, once it gets to CSI's lab, is I am usually the one who performs microscopy. So looking at the sample underneath the microscope, this is the only tried and true way of identifying the bloom and knowing what is causing it. There are other microorganisms and other natural environmental things that can happen that look very much like a bloom. Right now, pollen is in abundance in the environment, and I've received 2 pollen pictures from concerned folks this week. Looks very much like a HAB in the water, but it is not. And so I look at the sample under a scope, and I usually see a picture of what is shown here. Shown in this image are our 2 most common genus of cyanobacteria that we detect on Cayuga Lake. So the one that's all whirly swirly is called Dolichospermum. If you were to zoom in a little bit more and kind of stretch it out, it looks like a pearl necklace. There's like cells linked together in a straight line. And this is a colony. This isn't a singular cyanobacterial cell. This is a colony of many. And then those other larger clumps are microcystis. So we only identify to the genus level, not to the full species level. 1, because I'm not an expert in cyanobacteria identification. 2, it's not necessary for the purposes that we need. We just need to know, is there cyanobacteria in this or not? And we don't need to know the species. I would love to get more experience in identifying cyanobacteria under the microscope. My background is in wildlife biology, though, and I'm used to counting birds, doing bird surveys, and, looking at things above water that you can see with your naked eye. And this was a big pivot for me in my career, but really, really enjoyable. And I hope, as the years go on, that I can keep learning more and more. But for the purposes of our program, we only identify to the genus level. Then, assuming that the sample passes that first round, and yes, this is a harmful algal bloom that contains cyanobacteria, we do two different analyses. One is chlorophyll A, and this is looking at the relative mass of the bloom. The results are not 100% representative of cyanobacteria. So cyanobacteria photosynthesize. They're a little tricky. Most bacteria do not photosynthesize. The term harmful algal bloom comes from the fact that early scientists thought that it was an algae causing the bloom because algae photosynthesizes and is green. Chlorophyll A is the green pigment found in plants and all things green. So when we're trying to get the relative, the mass of the bloom, there can be other organisms mixed in that contain chlorophyll A, like phytoplanktons and other algae. But in a bloom, the predominant organism are trillions and trillions and trillions of cells of cyanobacteria. So we can make an educated assumption that most of the mass is from cyanobacteria. Now, this is the really important analysis, microcystin. So a moment ago, I mentioned microcystis, ending in IS. That is the genus of cyanobacteria. Microcystin is a toxin, a cyanotoxin, created by multiple different genus of cyanobacteria, not just microcystis. For the first year of my time with CSI, I could not keep them straight. Microcystin, microcystis. And we had an intern who, God bless interns, she said, well, microcystin is the toxin and they both end in IN. And I said, you are brilliant. And so that is how I remember it. But microcystin has a lot of health implications for humans and animals. This is a hepatotoxin or a liver toxin that can really affect people. I don't know what the true number of people who source drinking water is from the Finger Lakes because I've gotten variety of numbers, but one consistent number I see is 2 million folks get their drinking water from the Finger Lakes. And that is not only through municipal intakes, like at Bolton Point on Cayuga Lake or the Seneca Falls, town of Seneca Falls, also on Cayuga Lake, but perhaps some of you are in this situation. Many, many people source their own drinking water via a shoreline or beach well from the Finger Lakes and may not have an adequate treatment system in place because dealing with microcystin and cyanobacteria is pretty tricky, and UV or reverse osmosis, chlorine, like not one singular thing will tackle this issue. It's pretty dynamic, and this is why we monitor for this. There's a drinking water limit set by the New York State Department of Health, as well as a recreation limit, which I'll explain in just a moment here. So what we learned from 2025 was that 88% of our samples had greater than 4.0 micrograms per liter. That's probably a very obtuse, abstract number for you, but I'll just kind of qualify it a little bit. So the New York State Department of Health has determined that lesser than or equal to, or lesser than 0.3 micrograms per liter of microcystin detected in drinking water is considered safe. If it's 0.3 or greater, it is unsafe for drinking water. Now, for recreation, if you're swimming, if you're boating, recreating in some way, coming into contact with the water, and I see a typo, Alyssa, the contact limit is a bit higher at 4.0 micrograms per liter. This still might not mean much to you. What is a microgram? I don't know. It's a tiny little unit of measurement. But I'll just share with you that one of our samples collected last year at Sheldrake Point had over 20,000 micrograms per liter of microcystin in it. So that would have been a very harmful bloom to come into contact with. And you don't just have to consume tainted water to experience effects of microcystin. If you get it on your skin, like you're wading in the water, you're fishing, it can cause dermatitis, rashes. You can also breathe it in when it's vaporized. So imagine tubing or water skiing behind a boat, catching wave spray on a very windy day. Perhaps you source your water from the lake and you turn on a shower and now the steam is aerosolizing it. So it's not just, oh, I went swimming and got some on my mouth by accident. There's lots of different ways that you can become or come into contact with this toxin. So to zoom out a little bit and look at all of the data over the years, 70% greater than 4.0 micrograms per liter. But I combined some of the categories here, and over from 2018 to 2025, 81% of our samples came back yielding results that were unsafe for drinking water and recreation. So even when we reduced the number of samples that we were collecting and analyzing, the trend still showed that well over half of the samples showed a toxicity that were unhealthy or unsafe for recreation and or drinking water contact. I was getting ahead of myself again. So one of the, I don't want to say critique, but pushback or questions that we get is, why are we still sampling? Why are we still collecting samples? What's the point? We know HABs are bad and they're everywhere. Well, this data is really useful, and it might not be used right in this moment, but as things develop and legislation changes, having certified laboratory analysis data that was collected according to a protocol will be really helpful in setting different guidances and use in a multitude of ways that I can't even fathom at this moment. But we collect data because if we don't collect data and we have nothing to say for the year, does that mean there's no issues with harmful algal blooms, or we just didn't document it? So while we have hardy and dedicated volunteers willing to go out and monitor the shore and collect samples when asked to, we will continue collecting data in this more strategic modified approach. So looking ahead to 2026, we've already had our first bloom, as I mentioned. A longtime seasoned volunteer texted me in late April, and I just saw the notification come across the main screen of my phone at first, and I thought she was just saying, "Hi, Alyssa, like looking forward to getting, back into the swing of things and whatever." And then I opened the text and read the whole thing, and she was letting me know she had a bloom on her shoreline. She was located just south of Harris Park. This is a frequently, she is a very well-seasoned volunteer. She regularly reports into the double digits every year. And it's shallow. The fetch of the lake, or the direction that wind is primarily blowing, it blows a lot of stuff to the north, eastern corner of the lake. The land use is a bit more agricultural there. So it did not surprise me in the least where the HAB was seen. It was just how early. It was a very sunny day, but it was very cold. It was very cold. And every time I think we know a little bit more about this phenomenon, the cyanobacteria kind of keeps us in check and says, well, I'm going to show up when it's really cold and you're not expecting me yet. You're not ready. I started our volunteer program a whole month earlier this year. Volunteers began monitoring on June 1st. In years past, it's always started on July 1st. But I wanted to get ahead of the game and make sure people were ready to go. And the cyanobacteria must have heard that and said, well, we're going to start even earlier then. So luckily, this volunteer was willing to collect a sample and drive it all the way down to Ithaca from Cayuga. We didn't do analysis beyond microscopy on it, but because it was so early, I mean, the photo was classic for a harmful algal bloom, but we wanted to just make sure. And sure enough, it was full of microcystis. So this is a screen grab from our database. We have a water quality database that is free, open access to anyone who wishes to access it and learn about all sorts of water quality parameters, not just harmful algal blooms. And this is a useful tool. Another question that came in during registration was, what is the HAB history at Stewart Park? So surprisingly, it is not one of the areas where, looking back at our database and reports, there have been a lot of harmful algal blooms reported, maybe just a couple over the years. Now, that doesn't mean that there wasn't a HAB there and someone missed it, but it's not a high frequency part of the lake. It is very shallow, it is very weedy. Of course, blooms can happen anywhere on the lake at any time, but it is, when I think of hotspots on the lake, Stewart Park is not one of them. Thankfully, we've had no additional harmful algal blooms reported since. Well, excuse me, I did have one reported last week, but it was very, very, very sparse. It was hard to tell from the photos, so it went unconfirmed. But other than that, we've not had anything reported. Our volunteers began monitoring last week and will continue through the end of October. Usually our end of season is September 30th, but I extended it by a month on both sides this year. And as I mentioned earlier, we are maintaining that strategic approach for sample collection at these 15 priority zones. All of them have coverage except for Mudlock. I am still hopefully going to find someone who's willing to monitor once a week and collect samples if they see any. And we've also just had a bit of a logistical change. Ithaca Yacht Club is no longer going to be a site monitored, and so I need to go back to Tompkins County Whole Health and figure out another site that they'd like monitored and samples collected at. So this is a very shortened version of my presentation, but thank you all so much. I'd be happy to answer some questions. Please take down my information if you want to send me an email or call me at the lab. Thank you all again. Thank you to Liz and the network for hosting. And again, to all the volunteers for putting so many hours into looking at the lake and documenting what they see.
Awesome. Thank you so much, Alyssa. That's a lot of good updates for us about the program and what's happening out there on the lake last year and this spring as well. It looks like it's 8 o'clock, so I want to just check in and see if there's one pressing question for Alyssa before we wrap up our program.
I see.
I've got two of my board members. So, okay, I did see Keith's hand first. Sorry, Dave.
Yeah, and now you embarrass me. I don't know how pressing it is.
Sorry.
But I am curious. You know, well, one is to clarify it is true that the reports for blooms are individual reports on blooms, and that's not the right way to ask. The number of blooms reported are the number of blooms and not individual reports. So if 2 or 3 or 4 people report on a bloom, that's one report. Is that correct? I think it is, right?
Kind of, sort of. So it's really difficult to determine what is a harmful algal bloom, a singular, like where is the line of delineation? Is it where our eyes stop seeing, that pea soup green color in the water?
Gotcha.
Or are there still potential toxins that we can't see that would be considered a bloom status? But so these are reports. Reports do not equal blooms. If we all were standing together on the lake and we all reported the same thing, I don't know how many people we have in here now, but earlier it was like 30. Would that mean we have 30 blooms? No, that's 30 reports. I try my hardest to tease out individual reports, but in the case of the shoreline wide on the Western Shore event that happened last year, linking them all together to kind of, even though some of them were pretty close together, gave a big picture, because on our database, we can't draw polygons. It's only XY coordinates. So showing as best we can, the spatial coverage of these things we do that through accepting reports that might be representing the same bloom. But so yeah, we would need some round the clock live water quality testing equipment to really tell us about minute parameters of the water, which is dynamic and changing constantly with a current, with a wind, a breeze, a boat driving by. It's really hard to define a singular bloom. So
Yeah. Okay. Good. Good. No, that's helpful. Then that kind of leads to my second question is one of the not criticisms, but one of the things that I've heard that, and I'm sure we all have over the years, to denigrate or minimize the extent of the blooms is that the increase in the number is more a function of the number of eyes looking out for the blooms than it is a real increase
sure.
in bloom activity. And I'm sure you guys have talked about it and investigated it and maybe modeled it. So what have you done to kind of address that issue, that it's not simply the number of eyes on
Yeah.
the lake, but it is in fact an increase in the number of blooms?
Yeah, that's a hard question to answer because how the program was ran and the number of volunteers who were engaged in 2018 is different than today. And over the years, many people, or more and more people are becoming more in tune that HABs exist,
Yeah.
what they are. They're encouraged or not emboldened, but they're encouraged to report what they see. So do we have more HABs occurring more frequently and covering more area of the lake? Probably. We also have a lot more people looking.
Yeah.
So it's kind of hard to compare year to year, but
Right. Yeah.
I
Okay.
think
No,
two
that's
things
good.
are going on.
Yeah, I understand that. And certainly anecdotally, there are more blooms. I mean, the anyone that pays attention to the water, knows anecdotally at least. So
Yeah.
yeah, no, thank you. That's very helpful.
Yeah.
Thanks.
And some of my greatest anecdotal tidbits come from volunteers who have lived on the lake for 30 years and can tell me what it used to be like when their kids were little. And they never had to deal with this, never had to worry about this, never saw this sort of thing before. And so it's not maybe scientific, but it's still very relevant and helpful information to have that context.
Yeah, no, exactly. Thank you very much.
Thank you.
Great. So folks, we're pushing well past 8 at this point. So I'm gonna suggest that we wrap up the program. If you do still have questions, and I see some have come in and there's some good chat happening over in that window, please feel free, and you're encouraged to follow up with Alyssa because she'll be thinking about HABs all summer long and we'll be happy to talk to you. I just also want to note that Cayuga Lake Watershed Network has begun publishing our HABs newsletter, which comes out during the summer. We do this weekly and have sent our first kickoff newsletter already for the start of the monitoring season, and we'll resume the weekly end of week Friday summaries as soon as we start getting confirmed blooms again. So if you haven't subscribed to that already, you can do that on our website, cayugalake.org. And it's a great way to get a sense of what the trends are week to week throughout the summer as you're planning your visits to the lake and staying attentive. And then there's a lot of additional information about harmful algal blooms, both our program and larger trends and things that come out from the state. So yeah, thanks so much, Alyssa, again for that great update. And we'll look forward to hearing from you more. Thank you to Emily again, and to all of you for joining this evening. I hope you have a lovely evening and stay in touch because we'll have more events this summer. And I look forward to seeing many of you in person. Good night. Thank you.
Thank you, everyone.
Yeah.